All right, good morning, everybody, and happy Friday. Welcome to the Friday Department of Biomedical Informatics and Data Science PowerTalks seminar series. We have a really great crowd here. I'm very happy to see all of you, and I'm really happy to introduce today's speaker, Dr. GQ Zhang. He's a professor, distinguished chair of digital innovation in the Department of Neurology, also VP of Chief Data Scientist, Office of Data Science. This is co-director of Texas Institute for Restorative Neurotechnologies and professor of distinguished chair of neurology. Also has appointments in medicine, biomedical informatics, and public health at the University of Texas Health Science Center in Houston. I and also other of our faculty know him well. Prior to joining UT Health, he was professor of internal medicine and computer science and inaugural director for the Institute of Biomedical Informatics and associate director for their CTSA at University of Kentucky, and spent much of his career at Case Western Reserve University, where he was division chief of medical informatics. And he received his Ph.D. from the University of Cambridge in computer science. His focus has been on cutting-edge computer science and information methodology to address big data challenges and bioinformatics challenges, especially through the querying information retrieval, data management, focusing on the human data interaction with data, all the support of clinical research, most recently in epilepsy and in sleep disorders. So I'm happy to introduce him. NIMP, Neuroanatomy-Anchored Information Management Platform for Collaborative BICAN Data Generation, Dr. GQ Zhang.
Thank you, Amy, for the kind introduction, and thanks everybody for joining this talk. And it's great to see friends and colleagues here at UIB. I actually visited UIB a couple of times on different occasions. So it was, there's been, because of COVID, less of the physical travel, but it's at least great to get on virtual meeting like this. So what I'm going to do is, well, for those who don't know, the Texas Medical Center is actually a largest concentration of medical healthcare and research facilities that includes Methodist, MD Anderson, Rice University actually was on the adjacent campus, and things like that. So it's a hub of healthcare and medical academic medical center next to the heart of Houston. And so thus, before I actually get to the content of the talk, I was fortunate to have many long-standing collaborators and a few of the fundings from various federal agencies, mostly from NIH and NSF, for various kinds of efforts, including those reported here. I want to get the acknowledgement out of the way, but also this talk, as you can see, is already kind of very heavily platform, kind of innovation driven. So there's less of a commercial conflict of interest to declare. So I just skipped that slide. But I want to, before getting to the specific Brain Initiative project, I actually would like to briefly talk about what kept me busy, because I want to keep you, some of you, updated on the activities after I moved to Houston. One of the topics that kept me busy was actually to publish this research monograph, actually acknowledging Jim Semino's guidance through various stages of this former methods, former concept analysis approach to analysis of biomedical ontologies. So this is essentially a collection and a reorganization and synthesis of about 30 publications around this area with my co-authors and actually other collaborators, including Olivia Bodenreiter So published last year. For those who are interested in, please take a look. And the other topic that's ongoing and that's also keeping me busy is the notion of logic-based phenotyping. We already know the value of real-world data, and that's determined by determinant of health, including social determinant of health. But those data are going to be useful only when we can put some structure, put some annotation meaning to it, and be able to query subgroups of the data in meaningful ways. Current systems, such as typical data warehouses, OMOP and I2P2, emerge, those kind of effort focuses on underlying data model and some core interfaces. But in the entire biomedicine, I would say, there's a lack of rigorous logical approach to define the human phenotype or other kind of phenotypes. So the example here is in intracerebral hemorrhage started by heart attack. It's a typical kind of temporal description. But if you can actually formulate this using ICD codes and the temporal operator B started by, that's actually defined in Allen's algebra. And the purpose is actually to elevate it to rigorous logical level. So we started this work and it's ongoing, and we're expanding based on the earlier work, temporal cohort logic, that's actually reported at the AMIA 2 years ago. So these are two things that I'm not going to talk more, but that has been keeping me busy quite some time and some ongoing effort. As for the Brain Initiative, I would like to quickly mention a similar paradigm, similar kind of situation, and then switch to the Brain Initiative effort, because these two efforts share the similar kind of underlying system architecture, actually. So the clinical research data ecosystem, if we want to leverage clinical research data for research, that's specifically EHR that's generated at the point of care, those kind of data registries and data warehouses I talked about have typically involved the ETL, data harmonization, and move to enterprise data warehouses, and then to disease registries, and then interfaces are exposed to investigators and users to query the data and generate insight that hopefully sometime in the future can feed back to the patient care setting, feedback to the EHR as, for example, AI plugins and things like that. So the top is a typical standard kind of approach. But as everybody knows, the steps in between are actually lengthy and quite costly in most cases, because it's heavily manual, personal kind of effort required to database, import and export data, transforming data and things like that, converting data to standard format and things like that. We are here, it's one of my long-standing interest is to have a system called an epitome kind of system, to-me system, tracking and optimized management system in the middle that's actually overlaps with EHR and overlaps with disease registries, which may have additional data that's not contained in the EHR, but needs to be linked. So the idea is that at the point of care, once data is generated, it should only be generated once. It should not be exported or captured a secondary time or more times. It should be reusable immediately if possible. And so that's kind of overlapping Venn diagram you see below. That's the direction and I'm have been trying to move forward with what's called an epitome effort. And I actually have written one of the opinion piece on bespoke EHR that could be leveraged to address physician burnout issues, and addressing in a way that's one specialty at a time, because the bespoke EHR is designed for clinical specialty dependent ways. There are some guiding principles how it should be designed, what kind of value proposition we should have, and we should actually definitely leverage the latest development in computer science, and not constrained by what we already have. And also it should feed back into the learning healthcare system and generate high quality data to facilitate the research. So this is, as you can see, this diagram here, it's a main kind of architectural diagram. And this epitome system, we were able to incorporate many of the key clinical care functionalities at the point of the care, where those documentation is done in a structured way using domain ontology as much as possible. But that domain ontology supports both the data capturing, menu-driven forms, as well as once data is captured, the ontology itself can also support research queries. So there's no data extraction involved in moving the data generated at the point of care to support research. And there's no data warehousing, enterprise data warehousing involved either in moving data out of a system and then ingest into a different system. We actually have a JMIR report on some of the preliminary results associated with this system. And so here we actually design what's called a combinatorial dashboard, where each dot is a step in clinical care, and different color codes reflect different status. Green stands for completed, yellow means it's scheduled, and the white means no information. And so the entire clinical care steps, and there are different kinds, can be captured by the system. And within a couple of click of button, you can actually filter exactly what patient groups are have performed which kind of tasks and are ready to be scheduled for what's next, and follow up and things like that, such as after procedure, assessment, and things like that. So the entire patient journey is captured using this combinatorial dashboard in the system. And the interface with EHR, and we actually have experience with interface with Cerner, Allscripts, and currently we are interfaced with Epic, actually 2 Epic instances pretty soon from 2 different healthcare systems. And so on the back end is the HR7 and messaging system, and that makes sure all the patients that touches upon epilepsy monitoring unit will be automatically having their messages forwarded to our system. And then if the patient doesn't exist, it will be created. If it's already existing, then their scheduling will be populated in our system. And then all the documentation of care happens within our system with pushback of daily progress notes and billing, and all of those documentation happens from push button using our interface. But then those documents are automatically routed to the parent EMR and stored in the right encounter settings. So here's a screenshot of what the ontology-driven data capture looks like. So in terms of annotation of seizure, and Also, after it's captured, we actually have the same ontology that actually drives the query interface, and the query interface results in the patient list that satisfy the query criteria, but also automatically linked to all sorts of multimodality patient specific patient information and clinical history and things like that. So this is a faceted query system because we can actually bring in one ontological term at a time and then fix the variables associated with those terms to determine the query widget and criteria, and then generate the specific content. And there's another piece that talks about specific ideas of faceted query and how it works on the based on ontology-driven way. There are requirements on the domain ontology in terms of properties to achieve the effectiveness, coverage, and precision recall of the query system. But those actually ties back to ontology quality assurance using FCA as well. So the idea is here is that actually the system is light on data management part. Once system is set up, the movement of data and the information happens seamlessly through the interfaces. And actually, there's a dashboard for management, for individual physicians' performance, and things like that for billing. All of those are actually incorporated in the system. And in that publication, we did a small survey and on different aspects and to see if it actually helped the physician burnout. And we're glad to see that basically the epilepsy monitoring unit, epilepsy division, the physicians were involved using this, and we have total of about 200 users, and 25 now are actually clinical attendees. They are happy with the system and then wanted to work with configuration of whatever EMR updates are given and help us charting the EHR domain as well. And so it's going to be staying around, and we're generalizing this approach to other specialties as we speak, as effort and time permits. And so that's the epitome system, basically a bespoke HR for clinical care at the point of care. Now I move to the BRAIN initiative, which uses a similar kind of design architecture, this eager bird kind of form. The BRAIN initiative, for those who are not so familiar with, actually started about a decade ago with Obama administration, and we're entering the third 5-year phase for the what's called BICAN Initiative Brain Atlas, Brain Initiative Cell Atlas Network program. And blocked here, this diagram shows various different pieces of funding. Each block is an NIH award. There's different mechanisms that's involved, U1, UM1. Each UM1 on top, there are 4 of them, is basically a large center such as CTSA scale. And then we have the U24, 4 of them serving as the coordinating Center for Data Informatics and archiving effort. And so you'll see 2 Zhangs, actually. Only one Zhang for the U24 is me. The other Zhang on the lower right is somebody else. So don't be mistaken by that. So as you can see, this is a highly complex data ecosystem from informatics point of view. It's generating de novo data. And also highlighted here, we have the specimen on the left-hand side. We have the sequencing on the right-hand side. These are 2 major modes of operation that requires a lot of attention and effort. And from the very first design, we have the QB Brain Specimen Portal on the top left and sequencing portal on the top right to be the core kind of data infrastructure required to move this BICAN program forward. And the complexity and scope of the program can be seen from this diagram by NIH Program Officer Yong Yao, assimilating all the awarded applications for relevant tissue and assay generation effort. So you can see we're interested in the entire spectrum of different developmental stages, from pre-birth to infancy, childhood, adolescent, and adulthood, and also different anatomical regions in the brain using different technologies, and actually across different species as well. There's many different assays used on the right column that are integrating the brain tissue in different ways, brain samples in different ways. So you can imagine the complexity of the data footprint that's generated de novo from this context. So we're facing challenges in different ways. One of the requirements actually is to ensure fairness at the point of data generation and not at the point of data curation. So data is already there. We annotate and curate to make it more fair. Fairness is a requirement at the point of generation. It's not an afterthought. It's by design. Second is that actually when it comes to sharing human brain specimen, because of the scarcity and the preciousness of those, because no live human will have, no healthy human will have their brain donated to cut into pieces for research, right? It's either from the surgery, but here, because we're interested in atlas, so we need to sample all the brain regions. So it's the procurement of human donors and different brain labs that are be shared by those projects across the BICAN, so that the tissue actually will be sent from one place to another, and it's highly unlikely that one single site has all the tissues that they need for their study. So this is very different from the prior 5 years of the main mode of focus, which is on the mouse model, where mouse are bred into their individual labs locally, and then their brains are harvested for research locally without having the need to send them around from different places. So for this, we need to track the tissue usage. We track the resources and data generated in each step of the way. We have a dozen data modalities from different assays, and a standard operation procedure needs to be established to ensure the tissue quality, the donor kind of sort of neurotypicalness, because we don't want to have the, so this is designed to be a reference brain atlas, and so it's supposed to be quote healthy, but of course nobody is perfect healthy in all the ways. And then we need to ensure fairness, we need to have standardized metadata to capture to inform the data capturing process. But then the metadata standards may not already exist, and we need to build it while we're capturing the data. So this is building the airplane while flying it kind of situation, and requires very rapid turnaround of the outcome, and using continued iterative refinement as the key strategy. And there are different systems, because this is the third 5-year period. There are already prior systems in place, and we need to leverage, for example, those data archives. So any system that's created new needs to be able to seamlessly interface with existing systems, and that can be a challenge because different systems developed at a different time, uses different technology, has different requirements, and have different design philosophies as well. So it's not the same kind of uniform requirement that we can enforce. We have to deal with what we have. And then collaboration among the stakeholders with different background and value proposition, it increases the complexity in team size. For those who are familiar with CTSA, team size is essential ingredient for CTSA hub and the CTSA sites. And so here we need to be able to coordinate based on team size, the activities and efforts. But by actually compressed milestone instead of yearly milestone, we have a quarterly milestone so that NIH program can monitor the progress. The entire consortium can actually address bottlenecks, challenges in a timely manner by focusing effort and attention to areas that needs attention. And for us, the data versioning and expansion are actually a routine mode of operation because we're building things, and so data captured in a prior version of the system needs to be migrated to a new version with expanded metadata, additional fields, additional behavior, and things like that, and additional data archives that will be depositing those. So it's a constant moving target. And so here's the example. If I have time at the end, I can show the web portal a little bit to just show what it includes. So this is the actual screenshot of human brain slab. And there's metadata associated with this particular slab in terms of its orientation, what's before, what's after, thickness, and tissue quality conditions, and things like that. And what you see two polygons. One is dotted line, that's the red color on top of the tissue. The other is solid line. So the dotted line means that the investigator wants to request this area of the tissue. The solid line is when the tissue request is being fulfilled, there's adjustment on how the tissue is actually being cut. And so that we have an accurate reflection and real-time almost reflection of what remaining tissues are available for other people to use. You'll also see some colored dots, round dots. These are actually annotations with the Allen Brain Atlas and with respect to individual slices. So we use Allen's 3,000-ish brain structure labelings as a way to tag the slabs. And then that will facilitate not only the region of interest that's dissected, but also later on query particular regions of coordinated sampling plan, how many studies are actually using tissue from this particular structure and things like that. This is actually going to be across all species and developmental stages as we move forward. So to tame this kind of challenge, this is actually written in my proposal from the very beginning, is that we need to have a view of seeing the progression of this data to information to knowledge. We're not talking about the wisdom at this particular context, but how data gets moved from raw data to more standardized, more meaningful interpretable data that's actually in the end on top feed into the knowledge brain cell knowledge base. That's one of the outcomes of the study. And on the soft side, our way of strategy of addressing, and this is our working philosophy or mode of operation, guiding principle, is success is determined by products and tools, in our case, delivered to the broader community and not by individual achievements. So this really needs to require us to have a broader view of what it means when any component produces something, how does it help other BICAN components moving forward? And this is actually early on the diagram of different existing pieces and new pieces, how they interact. We have the what's called IMS on the below, and MBB Neuro Biobank from NIH that has been in existence for over probably 2 decades. And IMS is basically NeuroBiobank's information management system. And then we have BICAN Investigator in the step 1, request tissue through our portal, Specimen Portal, and then those tissue will be either forwarded to the NeuroBiobank or UM1 awardees institutions where they also have some human brain tissue. And then that tissue gets libraried and then put into 2 kind of common sequencing centers, Broad and NYGC. All the sequencing effort are performed at these 2 sequencing centers. We call them sequencing cores. But the entire step of the sequencing library to the aliquot, to FASTQ file generation, deposition to NEMO, are tracked in the SeqLib Portal in the middle there. And we have dashboards for different stakeholders to look into the status of their progress about their task at hand and what they're interested in. So in terms of dashboards, if you look at below, we have quite a few. We have investigator dashboard, IMS dashboard, MBB dashboard, SeqCore dashboard, NEMO dashboard, EM1 dashboard, NIH dashboard. And on top of that, we are refining and exposing APIs so that different stakeholders can actually grab different data, including metadata definition from our portals anytime they're needed. So there are different kinds of flows that's involved in this diagram. There's information flow. There's material transfer. That's tissue gets shipped from one place to another. There's actually generated data to different systems. That's yet another kind of flow through these different systems. And the kind of progression of activities are actually motivated this way. We have the UM1 projects that have scientific aims, operational priorities, and joint milestones. Those aims and priorities, milestones will drive what kind of a portal and archive functionality needs to stand up at what point in a just-in-time, mostly just-in-time fashion. And then those kind of information collected in the data archive and portals will feed to future data catalog and the entire BICAN dashboard, knowledge base, and things like that. So we have this progression from lower part informed by science to the middle part informed by data capture in portals, NIMP, our platform included, and then feedback to the public facing in the end. That's going to be in the second part of this BICAN award, that'll be so that other external investigators can already use the data that's generated from the BICAN initiative. And the purpose and guideline here is that as soon as data is generated and open accessible, it'll be put into the public domain for access. There's no requirement or strings attached. There's no requirement for the BICAN investigator to analyze the, to have an embargo period. So we analyze the first for 6 months or a year. There's no such steps in between. As long as data quality and annotation, interpretation information are all packaged in the same data releases, we will do that as soon as possible. That's the actual NIH BICAN mandate. And so the way we address some of those challenges, specifically those challenges with respect to sharing human brain tissue, who has what and doing what, where do tissue come from, what tissue generates what kind of data, where data are landed, deposited in the end, what metadata associated with the data and things like that, will be reflected in the portal, tracked in the portal. And I talked about different stakeholders have different value proposition. That's reflected in the kind of dashboard they want to be exposed to. And we need to use a role-based access control so that different people, different roles see different dashboard for their interests that they're in. And the entire portal will be web-based, a database backend, using existing and the legacy systems that will be API as the way to communicate and exchange information. The standard operating procedure sometimes do not exist, and the portal actually help inform the standardization of the process, tissue quality, data quality, and things like that. It also drives the terminology standardization in terms of minimum metadata and things like that, because it's driven by actual real-world usage. And so the strategies we adopted is prototype-driven portal requirement elicitation. So we have the demo, we have a working prototype, and then require further requirement from that. So it's not documentation oriented based kind of requirement, but it's actually live prototype based requirement elicitation. So a live interface is worth 1,000 words. That's the slogan we use. And prototype, so as I mentioned, the strategy is a deliberate strategy to use the prototype to drive the standard operating procedure as well as terminology development and standardization, and particularly in identify where are the metadata gaps, where are the terminology gaps, so that brings the ontology group to quick focus. One of the innovation of this our NIMP platform is that early on, from very beginning, we recognize the importance to have resource identifiers, what's called NHASH, that we used for other projects, for provenance documentation, resource tracking, and for enabling API calls. And this is so critical and serves as actually scaffolding backbone for the entire BICAN resource catalog. And we, of course, building it while using it, that means that we continuously refine and expand the functionalities with key stakeholders. And actually, NIH is one of the key stakeholders. They often periodically have new requirements for the portals. And on the back end, it's Ruby on Rails, as many of you probably who worked with me and chatted with me before knows. We use GitHub to both deposit code, but as well as managing the agile development process. We have testing, issue tracking, and we also release terminologies through the GitHub. We actually have a working and alive in continually expanded user guide that's refined over time as new features are being built. We actually, our team has over a decade of experience working together. That's we're in a fortunate position to do this. We have already done multiple systems, as I mentioned, the epitome included. So we're not here for the first time. Otherwise, it'll be daunting and overwhelming to undertake such task. We have reusable principles, reusable strategies, and reusable design components that we can rapidly adapt to the new situation. We also have team science experience and enjoyed working with the domain experts, and we're building new collaborations through this effort. And for example, we actually have established a very close working relationship with Allen Brain Institute, some of their investigators. And so here are remaining challenges and lessons learned. So you can quickly read through those. I want to, I should stop and leave about 10 minutes for QA. Is that okay?
Yeah,
Anyone?
but it's not a hard and fast. If you know, if this is if you feel like these things, anything you want to highlight, it's not. Yeah, I don't feel like it's a strict limit.
Okay. Yeah. So there are actually challenges in managing milestones, but milestones are there are dependencies, managing operational priorities, and there are competing interests, but the limited bandwidth. The system is almost like forever beta. There are always broken links because if you have new features that may interfere with the existing functionality, but we trust. I have confidence that the backbone architecture is what I call future-proof. We can actually build it on this solid foundation. And to actually increase communication and dissemination, we actually open up 3 times a week office hours. Anybody from BICAN can jump in and hop into our group meeting setting. And we usually have the first part of the meeting devoted to BICAN-related activities. And fair, it's really should not be a requirement. It should come naturally by doing the right thing, and doing things right. You automatically should achieve fairness. Again, this eager bird-like portal has this functional areas. That's data intake, show the data through dashboard, mediate different functionalities, and then interpret with data archives, and allow people to explore what kind of resources are in here. So I mentioned NHash and the tree diagram below. You can see how the main resource areas are actually having their identifiers generated using 2-letter prefix. Donor starts with DO, brain slab starts with SL, tissue TI, and things like that. And we use this randomized n-gram hashing. And how large is the n-gram is actually dependent. You can see the IDs are of different lengths. Depends on the estimated number of resources in that particular type of that particular type. For example, library aliquot, we accept quite a large number of library aliquot. Donor, we probably will have a limited number of donors, and at most in the thousand, the several hundreds. But so that we don't need a very large number of them. But the key property of the NHash is actually it uses provenance information as a way of encoding the code, generating the code. So actually, if there's dependencies on the parent, some snippets from the parent will be randomly extracted and used in generating the next identifier. So that once generated, the linkage is there solid and they're not easy to break. It's almost like a simplified blockchain technology in this setting. And once we have those, you can see this is a visual interface using Sankey diagram. We can actually track from a particular donor what kind of resources are generated from this particular donor all the way downstream. Or you can look upstream. I have this aliquot. How many donors are involved in tissue combined in generating this aliquot and library pool and things like that. And so this is the design of the landing page, how it looks like, and our actually go there live to the portal site to show what it looks like, and where at the same time I can entertain questions.
Yeah, thanks for that presentation. I actually just went to the BICAN website, and I saw, anybody could see things and download data and genomic data. So I thought that was pretty fascinating. What is the status of the sites? I have a question. Please feel free to ask your questions and enter the chat, or you can feel free to unmute yourself, because many of us are familiar. So what's the current status of the sites? Are you expanding? Or it's just it looked like there were a few, and the ones that had the grant that were in that grant. So because we obviously want to get lots of tissue from different places, not just from a limited number of places. So what's the status of that? Will that be expanded, or can you say more about that?
Yeah, it's a different technology. So we talked about the sequencing, and sequencing is basically operational through the two sequencing cores. But there are additional modalities, for example, Murphy's spatial transcriptomics is comes with a large data footprint, and CMU Pittsburgh have imaging data archive that will be linked to our system using APIs. So we're expanding in the data modality and linking additional data archive. And API is under constant refinement to support other activities. We actually also have new mode of data requests that needs to be accommodated. So there are many. So the portal is actually under constant redesign and refinement, and because of the different BICAN kind of needs. So I don't know if that answers your question. So.
Well, I'm glad to see that, I know you're expert in human interface with the data. So, I've always liked seeing your interfaces. I was thinking like the actual sources of the tissue specimens. So I think they're only coming from certain sites. Will that be expanded so that other sites can contribute their specimens and data?
Yeah, that's very welcome. I mean, they always short of specimen, specifically developmental, even pre-birth, kind of specimen. But so here listed are some of the contributing site. My name is a testing brain bank. It's for testing purposes, but the other ones are genuine contributors. And we always welcome additional contribution, but the tissue quality needs to be assessed so that it fits the purpose. That's the only requirement. But of course, we're always lacking. If UAB has a brain bank that can contribute to this, I mean, we'd be happy to connect it. And actually, within our institution, UT Health Neurosciences, we're repurposing this platform as our internal biorepository management engine. And so it's going to be custom tailored in some ways so that we can reuse our effort already spent here.
Yeah, thanks for that. Actually, let me.
Jin, you have a question? You see your hand.
Hi,
Oh yeah, Jin, go ahead.
Yeah, I'm so excited to hear your recent research here. So I have two questions, GQ. The first is about the scalability, because when you build up such a platform that's used by many users, one of the things come up is how do you scale up, right? How are you going to make your system available, not for a few users, but hundreds or even more users? So that's my first question. My second is about the user end, because if you think about the patient portal, many patients want to use the portal to read their data, and maybe they will provide the information. So your platform basically right now, I think it's for researchers or for the clinical side, for the clinicians, providers. What about the portal for users, for our patients? What would be the difference regarding the design principle? Thank you.
Yeah, thanks for the question. About the scalability, as you can see, we have so BICAN has a deliberate design so that heavy data footprint will actually be stored in data archives, which as CMUs, BIO, that's imaging data archive. They basically are constantly expanding their petabytes of storage, so we're not designed to be a primary data storage side. So other than that, in terms of number of users, there's really not that of much of a sort of resource scalability issue for us. And actually, people interface or use the system for uploading, downloading data most of the time. And these are mostly structured text and things like that. When API hits, we'll see. We deliberately also restrict the kind of API calls that can be permitted, because some of the API, if you completely open up and if you multiple API calls at the same time, that can slow down. So we're conscious about the performance, scalability of the system, but then so far it has not been an issue, and we're prepared to address it as if it arises. Your second question about the patient-facing portal, we have not focused on that domain, but of course for HRR, there's a My Chart. That's actually very well-designed kind of portal. We're not going to sort of do redundant job in those kind of domains, but any of the patient relevant information to the generated from the epitomy system is fed into Epic, and then Epic to patients through their patient-facing EHR. So that's an area that's we have not focused. So I can't say much about the experience there. But as a user of My Chart, I'm actually quite impressed with the immediacy and clarity of especially recent test results and things like that.
Other questions? Feel free to raise your hand, or any questions from the room, because you might not have access to the actual computer. I have some additional questions, and feel free to jump in. I'm just thinking like nuts and bolts. I don't have a lot. I'm not a tissue researcher or genomic researcher. I don't have a lot of experience with using biobanks or data biobanks and specimen banks. So say you had that interface, you don't have to show it, but where someone says, I want this area of, I'm interested in this area of the brain, and so I want to study that, and I'm making a request. Then am I only just getting sequence data from that? Or what if I want to do something with that actual tissue? Or is tissue being transferred, or is this really about sequence data?
It's as one of the earlier page, there's a dozen assays. Assay in the terms of biomedical, biological science area, that means that a mode of usage of the tissue. So some are, for sequencing, others spatial transcriptomics, and also Murfish is one of the latest technology developed by Xiaowei Zhang, I think, who's inventor, and she's a UM1 component. So those are the ones that you actually use in genes as a way of sort of overlay to the tissue in high precision and see which has been expressed and things like that. Gene panel, that's what they call. So there's a large variety of different data modalities. And for the first year, initially, we have been focused on the sequencing side, seeing basically understanding what kind of cells, their genetic profiles are existing in different brain anatomies. But it's expanding to many other kinds of data acquisition modalities. If I understand your questions.
So this is like a dry tissue biobank, like it's not a wet where I actually want that piece of tissue and get it in my lab and do some kind of assay on it that someone else didn't do, or it's only based on something that somebody already else assayed, and I'm getting their data. Just trying to see if it's like
Oh, it's dry. It's completely wet.
okay.
Yeah, it's completely wet, but our portals are tracking those activities. So you can actually request a wet frozen tissue from this portal, and the tissue gets shipped to you. You build in, usually people who request tissue, they already have some technology that they have a purpose, and including sequencing, right? So they go through the grinding barcoded cell sample process, put in the tube, and then ship to Broad. Broad, they sequences those and using our metadata and then deposit to NEMO. NEMO is the sequencing data archive. And then that investigators can access the FASTQ files and the BAM files after alignment through NEMO interface. So it's an entire data generation system from the acquisition of new donors, actually. Some of the brain banks are actually harvesting and using new donors from their site.
Other questions? We're kind of getting to the end here. Maybe you could say more about the metadata. So like I'm assuming there's metadata from that has, okay, it's from this person. I assume it's de-identified with these characteristics and these kind of diseases. I'm kind of interested in learning more about the metadata that comes with a specimen.
Yeah. So can you see the screen?
Yep.
So as part of the portal, there's a place called Resource Explorer. And this Resource Explorer can allow you to query donors, query brain slabs, visualize resource using Sankey diagram upstream or downstream with different depths, and then actually click and link to those rendered resources. The last item is terminology. So we actually have carefully documented, through the brain banks. The brain bank has their established donor metadata, field diagnosis, procedure, toxicity, pathology reports, and all of that. And so there's variable names from the source side, and each new variable that's used in the portal, we have n-hash identifiers generated from it. And there's a description. Then there's a BICAN, there's a separate ontology group that actually create a UUID for it. And then that's linked to external ontological terms if it exists. And also we define the value set associated with each variable, because that's mandatory for portal to be functional. You have this variable, you need to enter value, capture data, and what's the value, what's permissible value, and things like that. So here is the link that you can actually download the entire CSV of the terminology as used in the portal, the latest version. So we're updating that. So yeah, so but one of the interesting, maybe challenging area is that even though our portal uses Allen Brain Atlas, it's one of the brain anatomy ontologies, if you wish, but there are about 30 different versions of them. And the question becomes, how do you reconcile? How do you pick one versus the other? How do you harmonize them? And things like that. That's a potential interest and topic, but we don't have the bandwidth to actually work on those. To me, more kind of equally interesting, different kind
That's
of.
just a microcosm of the challenges we face in terminology in general. So
yeah.
Yeah, well, this is so fascinating. Maybe I should become a brain researcher. Anyway, any final thoughts, comments, questions? We're really over time now, so I need to let everybody go. But
yeah, thanks. Thanks for the invitation, and happy to follow up with any questions in any aspect. And if there's any topic we can work together, I'd be happy to engage.
yeah, you're well known to us, and you know us very well. We have all kind of collaborations in the past. Well, hearing well, we're over time, so thanks everybody for attending and also for helping with the people in the room also and on the Zoom. And thank you to Dr. Zhang for an enlightening presentation. And look forward to seeing you at the next Power Talk. Everybody have a great weekend. Thank you.
Thank you. Bye-bye.